A little bit story from my personal interest in platelet function. It started 10 years ago when I started my PhD thesis. I was evaluating platelets and searching for new markers which could explain inflammation and maybe the way to thrombosis. So this is the table, only one part of my thesis where I studied the flow cytometry parameters related to platelet function. And in my thesis we came with a conclusion that platelet function in men with dyslipidemia and with more risk factors, traditional risk factors for cardiovascular disease and metabolic syndrome, are really prone to increased reactivity. Increased platelet reactivity can be considered as an independent factor for cardiovascular disease. What's up to date? I was thinking what to tell you about platelet interactions and their role in thrombus formation, and my eyes caught up a really nice review paper on Nature series, and I will deal with some new things they describe about formation of platelets function, their heterogeneity, signaling and communication. And I will not discuss genetics at all. We will discuss what is the role of platelets in normal physiology, in hemorrhagic or thrombotic conditions, and of course the role in inflammatory disease processes. We very well know that platelets are formed in the bone marrow, megakaryocytes. They evolve from progenitor cells and live between osteoblasts. They can migrate through the bone to more vascularized places. They produce pseudopods, large pseudopods, into sinusoidal vessels, which later become platelets, as we know, in the peripheral bloodstream. What we very well know that there's a negative feedback of thrombopoietin, which makes platelets produce more. If platelet number decreases, there are signals for thrombopoietin to be produced. What is, well, not quite new, but very well understood today, that inflammatory processes might induce thrombopoietin secretion and consequently production of platelets. And one of those markers which is responsible for that, it is interleukin 6. What is the fate of platelets during their short lifetime of 7 to 10 days? Well, part of the platelets will become old platelets, and we call this process aging. During aging, there are certain biochemical processes that are happening. First, decellulation of glycoproteins, then platelets become prone to phagocytosis in the liver or spleen and other organs. There's this balance of back pro-apoptotic pathway with anti-apoptotic pathway, and there's also the third mechanism, which is named membrane ballooning, with the exposure of phosphatidylserine, which is also a trigger for phagocytes to consume platelets, aged platelets. During the lifetime of platelets, total RNA content decreases, and platelets become, as they are called, exhausted ones. But this is not the main way how platelets are removed from vasculature. There are other mechanisms like seclusion in spleen, consumption, deposition at the activated vessel wall, activation per se, and microvesiculation. And there is no consensus which way is dominant, and it is not really good understood yet. Well, platelets are really heterogeneous in structure and in function, as we will later see. In previous century, it was told already that larger and denser platelets have a greater metabolic potential than smaller ones. But to tell you the truth, heterogeneity of platelets is really high. For example, they differ in size, they differ in their content, called light or dense platelets. They differ in protein expression profile and the content of glycoproteins. And they are young and they are old. So giving this slide, we can tell that the 2 by the power of 4, we can have 16 different platelet populations in one single individual. Well, individual platelets can differ from each other, as told already, and to perform their classic functions in thrombosis and hemostasis, like adhesion, aggregation by activating glycoprotein 2B3A, secretion, and procoagulant activity. We have to keep in mind that not every population of platelets will give the extent of their signaling the same way. So it differs whether they are secreting platelets or not, whether they have activated integrins or not, whether they can carry a fibrin clot on their surface, and whether they can process with the membrane ballooning. Let's go through the main signaling mechanisms which are really related to interconnection between platelets together and the environment, and also with other cells in the bloodstream like leukocytes. So let's start with the main function of platelets in normal blood vessel. It is inhibition of procoagulant pathways. As told with the previous lecture with the professor, that prostaglandin and nitric oxide are the main mechanisms of inhibiting platelets from becoming procoagulant. Initiation of platelet activation is mainly carried out by 2 integrins who bind with collagen and von Willebrand factor. In the environment, secreted adenosine diphosphate and thromboxane, they stimulate P2I receptors. Then there's amplification of the signal with collagen and fibrinogen via glycoprotein 6 and CLEC2 receptors. Thrombin is really potent activator of platelet function via PAR1 and PAR4 receptors. There's a mechanism where platelets get higher affinity for fibrinogen in the thrombus formation mechanism. Platelets are able to secrete thromboxane A2, which is also, again, the potent activator of aggregation. This is a secretory function. A group of proteins called snares are responsible for this process. And the last one is a ballooning mechanism where protein degradation is happening. Salt and water is coming into the platelet, and phosphatidylserine is expressed on the surface. There are many factors which actually influence the platelet heterogeneity, and we have discussed already intrinsic factors like platelet size, volume, levels of membrane receptors, cytoplasmic, granular, or cytoskeletal proteins which are responsible for activation or inhibition, and the aging of platelets. And there are extrinsic factors which also influence how the thrombus is formed. So it's local availability of different agonists in the place where the platelet lives. the type of adhesive surface which is represented when the injury happens. What is local rheology? Is it a high shear or is it a low shear stress? And what are interactions with other platelets and other cells? So review paper gives the view that there are four main populations of platelets who perform their vital functions. So one population is aggregating platelets who have high affinity to fibrinogen via integrin alpha 2b beta 3. The second one is procoagulant platelets responsible for thrombin generation, the first thrombin molecules formed in the clot. Then there are coated platelets who have fibrinogen molecules all over the surface of the platelets and are responsible for keeping the structure of the thrombus. And there are secretory platelets who secrete active metabolites and proteins into the environment and then also take place in the thrombus formation. So looking at the processes step by step, resting platelets having signals on their own, they might connect to the subendothelium via two glycoproteins. Glycoprotein 1b responsible for connection with von Willebrand factor. Glycoprotein 6 is responsible for connection via collagen. Aggregating platelets then start aggregating. The small clot is formed and there already secretory platelets secrete their active substances. Later on, thrombin is generated more and more and the clot grows. Coated platelets, they save clot from outside inhibitors like nitric oxide and prostaglandin. And there is a really nice cooperation of secondary hemostasis and coagulation pathways which were pictured in the last presentation together in the platelet thrombus. So again, we have tissue factor who triggers the generation of thrombin. We have formation of procoagulant platelets and coated platelets, amplification of thrombin generation, activation of the intrinsic coagulation pathway via factor 12, and the expansion of fibrin network and the security from inhibition of platelet activation. And once more, I would like to emphasize that there are different conditions, therefore we have different clots. In arterial thrombosis, clots have more platelets on them, and we usually call those clots white clots, and the conditions are of high shear flow. In the venous thrombosis, endothelium might stay intact actually, and clot differs from the arterial one because it is with more red cells in the clot, and we call that clot the red one. Well, platelets communicate, as told already, and it is interesting that they are communicating bidirectionally. They communicate, platelets influence environment, and environment also influences the platelets. The main mechanisms which include influence on the environment are secretory function like alpha and delta granule secretion, lysosomal secretion. And there's two mechanisms which were previously less described. It is eicosanoid release and extracellular vesicle shedding. How environment influences platelets. Platelet response might be sensitization or desensitization to activation, might uptake plasma components or even cellular components, and it is one of interesting areas for research where platelets might uptake tumor cell information and carry it to other places. The way how platelets behave in our circulature is really balanced in the normal physiology, and platelets would never become secretory, aggregating, procoagulant, or coated ones because there are negative and positive primers which are in balance. And there's a theory that people with the risk factors for cardiovascular disease might have more positive priming factors and thrombosis might not form, but even in a small changes in the endothelium during atherosclerotic process might trigger fast platelet response, and then secretory platelets, aggregatory, procoagulant, and coated ones are generated. So positive primers include adrenaline, cholesterol, ECAM molecules, prostaglandins, soluble CD40, thrombopoietin, and von Willebrand factor. And negative primers are adenosine, insulin, nitric oxide, and prostaglandin, which acts on different receptors compared to positive priming. It is clearly known that platelets play really a huge role in vascular thromboinflammation, and this picture depicts that during the lifetime of a human, platelet coagulation activation is increased. And there are vascular changes during the lifetime. First, we have healthy vessel, then early atherosclerotic lesion grows, and it advances through age and might rupture or might have an erosion. And the effect of platelets onto the thromboinflammation is increasing during that lifetime. There are four main mechanisms in thrombosis for platelets. Two are classical ones, actually, like activation via collagen von Willebrand factor, the one, and the second with a tissue factor activation, thrombin generation, and then platelet activation. What I wanted to emphasize more that the two less known mechanisms like the C, which is ultra large von Willebrand factor molecules, which are really thrombogenic ones, attract platelets, and platelets might aggregate or even get away from the endothelium into the peripheral small blood vessels. The fourth one is related with inflammatory diseases caused by pathogens like viruses, bacteria, and here neutrophils play a very great role. Together with platelets, they make neutrophil extracellular traps, which get rid of pathogens very well. But again, platelets are activated and thrombosis might take place if balance is not in good condition. Well, knowing platelet functions and how platelets interact with secondary hemostasis proteins and other cells, we might draw a simplified scheme and point the places where the targets are for antithrombotic or anti-aggregatory treating targets. So up to date we have quite good range of anti-aggregatory and antithrombotic drugs which can be monitored in laboratories. The list of methods or assays here is not the final one, it's just an example that light transmission aggregometry, verified now, approved by FDA in USA, probably might be of first choice. There are other methods like whole blood aggregation and others. So we have cyclooxygenase 1 inhibitor, aspirin, or other non-steroidal anti-inflammatory drugs, P2I inhibitors, integrin alpha to B beta 3 inhibitor, and PAR1 inhibitor. But there are future potential targets which could be used together with the current antiaggregants. It is really interesting that direct factor 10A or thrombin inhibitors used together with P2I inhibitors show really good treatment modalities. And to conclude what I have said today, I have 5 messages I hope you will bring back home. That there are platelet populations with heterogeneity in formation, structure, properties, and that there are consequences of this heterogeneity for functional specialization. Important roles of platelets in thromboinflammatory processes in interaction with leukocytes and the vessel wall are being really good understood. There is evidence on how circulating or adherent platelets can communicate with their environment via different mechanisms, via lipids, bioactive peptides, proteins, RNA, extracellular vesicles. The response adaptation of platelets to their environment is taking place by negative or positive priming, and the development of new medications for the treatment of cardiovascular disease and related conditions is actually our future, I suppose. I really thank you for the attention, and I wish that there's health and peace to everyone.